Welcome to UGC series of lectures. In this lecture, I will discuss the most common structural chromosomal abnormalities, that is deletion and duplication, and will deliberate upon their origin and genetic consequences. Dear viewers, as you know that a deletion is a chromosomal alteration involving the loss of a segment of a chromosome which may be located anywhere along the chromosome. When a deletion occurs near one end, it is called terminal deletion. When it occurs from the interior of the chromosome, it is called intercalary deletion. It would be pertinent to mention that deletion was the first structural alteration detected by Bridges in 1917 from his genetic studies on the X chromosome of Drosophila. You also know that a chromosomal duplication is a repeated segment of the genetic material. Various kinds of duplications have been observed. If the duplicated sections are adjacent to the original, the process is known as tandem duplication. Whereas, if they are separated by non-duplicated regions, the duplication is said to be displaced. And if they are existing as independent fragments, then it is termed as extrachromosomal. As far as the origin of most chromosomal structural changes are concerned, in most of the cases, these changes are due to one or more breaks along the axis of the chromosome, followed by either the loss or rearrangement of genetic material. Chromosomal breakage occurs spontaneously in a low frequency, but the rate of breakage is increased relatively in a high frequency by several radiations, chemical agents, and by several viruses, for example, measles virus. In addition, some genes are also known to induce chromosome breakage. For example, activated dissociator gene of maize and other transposable elements of eukaryotes. Although the actual ends of chromosome known as telomeres do not readily fuse with newly created ends of broken chromosomes or with other telomeres. The ends produced at point of breakage are sticky and can rejoin with other broken ends. If the breakage and rejoining does not re-establish the original relationship, and if the alteration occurs in germplasm, the gametes will contain the structural rearrangement which is heritable. If the alteration is found in one homolog but not the other, the individual is said to be heterozygous for the aberration. In such cases, unusual but characteristic pairing configurations are formed during meiotic synapses or in somatic cells if chromosome pairing occurs. Now, let us take the origin of chromosomal deletions. As far as the terminal deletions are concerned, these are the simplest rearrangements involving only one breakpoint in one chromosome. The initial event produces two pieces of chromosome material, one with the centromere and one without. The latter, termed as a centric fragment, can't go through mitosis since it has no centromere and therefore lost. Intercalary deletions result from two breakpoints within a chromosome arm and loss of the intercalary segment between these breakpoints. Let us now discuss the origin of chromosomal duplications. Like deletions, duplications also arise as the result of chromosome breakage and reunion of the chromosome segment within homologous chromosome. As a result, one of the two homologs involved in the production of a duplication end up with a deficiency, while the other has a duplication for the concerned segment. Duplications may also arise due to unequal crossing over between synapsed chromosomes during meiosis. For example, tandem duplication of the chromosome region called 16A of X chromosome in Drosophila. Gametes containing the duplication produce boar offspring. Males carrying the boar duplication in a hemizygous state have severely reduced eyes. Heterozygous females have a boar and a normal chromosome have slightly reduced eyes. Replication error during meiosis also leads to chromosomal duplication. Dear viewers, the genetic effects of deletion depends on their size. A small deletion within a gene called as intragenic deletion inactivates the gene and has the same effect as other null mutation of that gene. If the homozygous null phenotype is viable, for example in human albinism, then the homozygous deletion will also be viable. In this lecture, I will discuss multigenic deletions, that are those deletions which remove 2 to several thousand genes. When a segment of a chromosome is absent, the genes normally situated in the missing segment are also absent. The consequences of the multigenic deletion depend on the genes or part of genes that have been removed. In diploid organisms, the effect may be lessened by the presence of a copy of the lost set of genes in the homologous chromosome. However, if the homolog contains recessive genes with drastic effects, the consequences can be severe. Several genetic consequences are produced due to chromosomal deletions. The first is that the most deletions produce recessive lethal effects. However, deletions may produce dominant lethal effects as well. The second is that the deletion of a segment on one homolog sometimes unmasks recessive alleles present on the other homolog, leading to their unexpected expression, because in such cases it seems that recessive alleles are showing dominance. The effect is called pseudodominance. The third is that the crossing over is completely absent in the deleted region of a chromosome, since this region is present in only one copy in the deletion heterozygotes. The fourth is that the deletion analysis makes it possible to compare a linkage map based on recombinant frequency with the chromosome map based on deletion mapping. By and large, where this comparison is made, the maps correspond a satisfying cytological endorsement of a purely genetic creation. Finally, many deletions produce a distinct observable morphological effect. Phenotypic effects of deletions depends on the size and location of deleted sequence on the genome. For instance, deletion that spans a centromere results in one acentric chromosome that will most likely be lost during cell division. Deletions can affect gene dosage and thus the resulting phenotype. Also, the larger the deletion, the more genes are likely to be involved, and the more drastic the resulting defect is likely to be. Dear viewers, let us now take the phenotypic effects caused due to deletions in different organisms one by one. Deletions in prokaryotes. Deletions are found in prokaryotes as well. For example, E. coli, Fog T4, Fog lambda, etc. In E. coli, deletions of up to 1% of the bacterial chromosome are known. In Fog lambda, however, up to 20% of the genome may be missing in some of the deletions. In these organisms, deletions may be detected by a loss of the function associated with the lack of recombination in the concerned region. Deletion in man. A number of human disorders are also caused by deletions of chromosome segments. In many cases, the abnormalities are found in heterozygous individuals. One human disorder caused by heterozygous deletions is cry-do-cat syndrome, named for the distinctive cry in children due to malformation of the larynx. Cry-do-cat syndrome, which occurs in roughly 1 in every 50,000 live births, results from a deletion on the short arm of chromosome 5, one of the larger human chromosome. Children with cry-do-cat syndrome are severely mentally retarded, have a number of physical abnormalities, and cry with a sound like the meow of a cat. Hence the name, which is French for cry of the cat. Another example is Prader-Willi syndrome, which results from heterozygosity for a deletion of part of the long arm of chromosome 15. Prader-Willi syndrome occurs at approximately the same frequency as that of cry-do-cat syndrome. Infants with this syndrome are weak because their sucking reflex is poor, making feeding difficult. As a result, growth is poor. The genetic basis of Prader-Willi syndrome is further complicated by the fact that the genetic region involved is also subjected to genome imprinting. That is, the phenotype differs depending upon which parent the deletion is inherited from. Deletions in Drosophila. One more phenotypic consequence of heterozygous deletion can be noted in mutant notch phenotype Drosophila. In these flies, the wings are notched on the posterior and lateral margins. Data from breeding studies indicate that the phenotype is controlled by an X-linked dominant mutation. Because heterozygous females have notched wings and transmit this allele to one half of their progeny. The mutation also appears to behave as a homozygous and hemizygous lethal because such females and males are never recovered. Deletions in plants. As far as the plants are concerned, an interesting difference between animals and plants is revealed by deletions. A male animal that is heterozygous for a deletion chromosome and a normal one produces functional sperm carrying each of the two chromosomes in approximately equal numbers. In other words, sperm seems to function to some extent regardless of their genetic content. However, in diploid plants, on the other hand, the pollen produced by a deletion heterozygote is of two types. Number one, functional pollen carrying the normal chromosome, and number two, non-functional pollen carrying the deficient homologue. Thus, pollen cell seems to be sensitive to changes in amount of chromosomal material. The three important genetic consequences of duplications are gene redundancy, phenotypic variation, and genetic variability. Gene redundancy. Although many gene products are not needed in every cell of an organism, Other gene products are known to be essential components of all cells. For example, ribosomal RNA must be present in ample quantity to support protein synthesis. The more metabolically active a cell is, the higher the demand for this molecule, which becomes a part of each ribosome. It is the norm for organisms to have multiple copies of genes coding for ribosomal RNA. Collectively, such DNA is called repetitive DNA or rDNA. And the general phenomena is called gene redundancy. For example, in the common intestinal bacterium E. coli, about 0.4% of the haploid genome consists of repetitive DNA. This is equivalent to 5 to 10 copies of the gene. In Drosophila, 0.3% of the haploid genome equivalent to 130 copies consists of repetitive DNA. Number 2, phenotypic variation. Most duplications have no phenotypic consequence, but large duplications of chromosomal material lead to gene imbalance that may be lethal to a zygote or even, in the case of plants, to the pollen or ovules that carry them. The classical example of phenotypic variation due to duplication in the Bar mutant on the X chromosome of Drosophila. This was first studied by Sturtevant and Morgan in the 1920s. These investigators illustrated that normal wild females have about 800 facets in each eye. Heterozygous females have about 350 facets, while homozygous females average only about 68 facets. Females are occasionally recovered with even fewer facets, that is 45, and are designated as double bar. Some 10 years later, Bridges and Muller compared the polytene X chromosome bedding pattern of the bar fly with that of the wild type fly. The Bridges-Muller studies revealed that one copy of region 16A of the X chromosome was present in the wild type flies and that this region was duplicated in bar flies and triplicated in double bar flies. These observations provided evidence that the bar phenotype is not the result of a simple chemical change in the gene, but is instead a duplication. The double bar condition originates as a result of unequal crossing over, which produces the triplicated 16A region. In this case, it was also observed that when the eye facet phenotypes of BB and BDB plus flies are compared, an average of 68 and 45 facets are found respectively. In both cases, there are two extra 16A regions. However, when the repeated segments are distributed on the same homologue instead of being positioned on the two homologues, the phenotype is more pronounced. Thus, the same amount of genetic information produces an altered phenotype depending on the position of the gene. Therefore, when a chromosome rearrangement involves no change in the amount of genetic material but only in the order of genes, the term position effect is used to describe any associated phenotypic alteration. Number 3, genetic variability. Duplications have played an important role in the evolution of gene families. An extra copy of a gene is free to evolve through the acquisition of mutation into a gene having a more specialized or different function. This process does not compromise the original function, since an intact copy of the original gene remains in the genome. Duplications are therefore raw material for evolution, and many examples of duplication and specialization are known. One example is the evolution of several forms of the globin chain of hemoglobin. Several globin genes arise from a single ancestral precursor, thus making individual genes available to take on specialized roles, with some genes becoming active during embryonic and fetal development, and others becoming active in the adult organism. Other examples include the immunologically important T cell receptors and antigens encoded by the major histocompatibility complex. I would like to close this lecture with this note that number 1, most chromosomal structural abnormality arises due to chromosome breakage and reunion. And number 2, chromosomal deletions have more genetic consequences than chromosomal duplication. Hope you might have understood the topic. Thank you.